Hey everyone, welcome to RNAHub. RNAHub is a website that hosts a specific type of RNA called 16S rRNA, which is highly conserved across many species. So this is important because researchers use it to classify organisms in the tree of life, and our own Carl Woese at UIUC discovered archaea using 16S RNA. So we get our data from Green Genes, which hosts all of this 16S RNA. And the first thing I'm going to show you is a basic insert. So what we do is we can put in the ID, which I'll just do 1, 2, 3, 4, 5. Common name, I'll do a fun name. Strain, sure. Sequence, ATGCGCG, and submit. So now that's going to be in our database. Going back to the text search, we're going to look for 1, 2, 3, 4, 5. It's going to pop up here with our fun name, and you'll see sequence, the length of the sequence, the name, and here we can edit it, so change it to something not fun, strain, no, and a new sequence, and update. And you'll see that it's all instantly updated right here with the new sequence. You can access publications as well, but we'll get to that in a little bit. So text sequence, so we'll search for one that's already in the database, 476. We see the name here, and we can see this is a much longer sequence, along with the accession number, strain, and some other information. So if we look, there is a publication. So we have this that we can look at and use for research. We can go on over to our first advanced function, which is called primer design. So primers are used in PCR and DNA sequencing, and you actually have to use specific calculations to make sure that they can be used. So like the Wallace algorithm outlines why you need GC content, melting temperature, and a certain length for them to work. So now we're going to go head over to sequence search. Actually, first I'll grab a sequence that exists just to make sure. So we'll copy and paste the 476 sequence back into the sequence search. Yep, right there. And yep, same thing pops up with all the information we saw before. Alright, next we're going to do our second advanced function, which is sequence alignment. Sequence alignment uses the Needleman-Wunsch algorithm, which we had to go and encode ourselves. Basically creating some data matrices that generate scores and then walking back and determining what the best possible scores are, which is actually a pretty interesting and fun task that we had to solve. Yeah, so you see that you align sequences, and this actually gives you an idea where mutations might be, or basically where insertions or deletions in the DNA sequences. So yeah, that's RNAHub, and I hope you guys enjoy.